Skeletal muscle is characterized by a hierarchy of smaller and smaller units. A skeletal muscle consists of a bundle of long fibers. Each fiber is a single cell with multiple nuclei. A muscle fiber is itself a bundle of smaller myofibrils. The myofibrils, in turn, are composed of two kinds of myofilaments, thin and thick. Thin filaments consist of two strands of actin and one strand of regulatory protein coiled around one another. Thick filaments are staggered arrays of myosin. Each repeating unit is a sarcomere, the basic contractile unit of the muscle. This arrangement of thick and thin filaments is the key to how the sarcomere and the whole muscle contracts. Contraction starts with the arrival of nerve impulses at a special site known as the neuromuscular junction. The impulses cause the release of these nerve terminals of acetylcholine, a neurotransmitter chemical, into the synaptic cleft. Acetylcholine then combines with the receptors on the sarcolemma, the muscle membrane, which then allows sodium ions to rush into the muscle cell.
This increase in sodium ions results in an upset called an action potential. The action potential spreads deep into the interior of the muscle fiber along infoldings of the plasma membrane called transverse or T-tubules. The T-tubules make close contact with the sarcoplasmic reticulum, SR. When the muscle fiber produces an action potential, the action potential opens calcium channels in the SR, allowing calcium to enter the cytosol, which is the fluid in the cell.
Calcium ions bind to the troponin complex. This shifts the tropomyosin and exposes the myosin binding sites.
At this point, the myosin head is bound to ATP. When the myosin head hydrolyzes ATP to ADP, it is then cocked back. With the myosin binding sites exposed, the myosin head is allowed to spring out of its cocked position and grab onto the actin, forming a myosin cross bridge. The myosin then pulls the actin toward the center of the sarcomere. Once the myosin head has made its pull, the binding of a new ATP molecule releases the myosin head from the actin. As many myosin heads alternately attach and detach, pulling the actin, the sarcomere shortens, which in turn shortens the muscle cell, causing muscle contraction. Contraction stops when the SR pumps calcium back out of the cytosol and tropomyosin again blocks the myosin binding sites on the thin filaments. At this point, the muscle relaxes.